Charlene McCord. I'm a postdoc at the University of Chicago. Today I'm excited to have the opportunity to share a bit of what I did for my thesis research, namely putting together a listoid phylogeny and then using it to investigate the diversification of triggerfish skulls. So first, a bit about my study critters. Phylogenetic is the superfamily within the very diverse order Heterodontiformes, all of which are probably most well known for their very strange cranial features. So Ballistidae and Monacanthidae, the two groups that you'll see listed here, make up the superfamily. They are sister families. However, there's a lot of notable differences between the two groups, a few of which I've listed here. Now, this disparity amongst very closely related organisms raises some interesting questions about the drivers and also the timing of these differences, and also sets a great stage for comparative evolutionary study, which was basically what my dissertation was all about. So today, two major parts to my talk. The first part will be actually how I put together a listoid phylogeny, formed a hypothesis of those relationships, and then the second part will be how I used that tree to anchor an investigation of triggerfish skull morphology. So I am certainly not the first person to have an interest in Heterodontiform phylogenetics. This is a particularly rich and diverse, fantastic literature. If you're interested in learning more, check out this recent review that was done by Matsur in 2015 for an exhaustive history of the group. However, explicit phylogenetic analysis of ballistoid fishes are virtually non-existent, with a notable exception of a fantastic paper that came out in 2013 from Santini, Sorensen, and Alfaro, which I will be coming back to in a few minutes to kind of compare our findings. So I was able to build upon this really fantastic phylogenetic history, add a few new genes, add a few new taxa. First and foremost, I mined GenBank, found a couple gene regions marked with asterisks here that have been commonly used in phylogenetic studies. And then I went ahead and sequenced 2 additional mitochondrial regions, 2 nuclear markers, as well as the growth factor BMP4, which we know has something to do with the ontogeny of both skull and tooth elements in a wide range of organisms. So regulatory genes overall are not commonly used in phylogenetic analyses, so we don't always have a great idea of what their phylogenetic utility is. But we thought that looking at ballistoid fishes, it might be a good idea to investigate the molecular evolution as well as the phylogenetic utility of regulatory gene known to influence cranial development in a group of fishes that's known for their weird craniums. So I sequenced everything, found the best model of sequence evolution for each region, put it into a super matrix, and then loaded up a partition branch run into Mr. Bayes, and got my Bayesian majority rule consensus tree here. So first and foremost, we are able to confirm the monophyly of the superfamily as well as both of the families that make it up. We also corroborate the long-standing sister-family relationship of these two groups. We also find good support for 3 clades within the triggerfish lineage and 4 clades within the filefish lineage. Now, within the ballistid radiation, we don't find a whole lot of differences between previously studied papers, so I'm going to spend a little bit more time looking at the filefish lineage because that's where some of the neater stuff came out. So again, only one other paper that's really explicitly looked at filefish phylogenetics. So here is the filefish portion of Santini et al., and this is what we came up with. So first and foremost, Oxymonicanthus longirostris, a particularly odd filefish. They found as sister to all the other filefishes, whereas I find it to be nested within what I call clade A. There's also some differences between Purveger, Stephanolebus, and Eleuteris in both of our trees, 3 major genera within the filefish lineage. Santini and colleagues found that Purveger and Stephanolebus are sister, whereas Eleuteris is a little bit more closely related to these more temperate fishes. Whereas I find Eleuteris and Stephanolebus are actually more closely related to one another than either is to Purveger, and also that none of these genera are super closely related to these temperate filefishes. You'll also notice this bottom part of both of our trees is where the least support is, and really it's the most interesting part of the biodiversity of these groups. The subtropical and also temperate regions of Australia and New Zealand is a hotspot that I really think we need to look into more, looking to the future of this particular group. So next, triggerfishes have a fantastic fossil record. So we were able to calibrate several nodes in our phylogeny, and also we were able to use, for the first time, a recently described filefish fossil. So then we ran all the stuff through BEAST. Nothing crazy here again. The time of diversification for each of these major nodes was within the confidence intervals of previously one or more previous studies. But just to kind of highlight a few things, we get a mid-Eocene origination for the superfamily. The listids were coming up in the Miocene, whereas Phylogenics are a little bit older, coming up in the Oligocene. We were able to kind of, using that filefish fossil, kind of shorten down, narrow those confidence intervals. So then, also, significant diversification within both families over the last 2 million years. The next thing we wanted to look at was molecular evolution. And so we investigated substitution patterns under a likelihood framework in HiFi. And essentially what we did for all of our protein coding regions is we mapped the synonymous and non-synonymous changes onto a pruned maximum posterior probability tree and essentially looked at the accumulation of these substitutions where they were happening. Overall, synonymous substitutions far more common than non-synonymous substitutions overall, except for a few branches and nodes, leading to DN-DS ratios usually less than 0.5, often less than 0.1, which is indicative of stabilizing selection. The accumulation of both types of substitutions was always more rapid at the base of the blastoid tree, lessening more towards the tips. However, in BMP4, we can see kind of a different sort of pattern here. It was quite often that within the filefishes, you would see a little bit more of a rapid accumulation of both synonymous and non-synonymous changes. But this was really a runaway sort of situation within this growth factor BMP4. We get 10 times the amount of substitution changes relative to the triggerfishes. Which is kind of interesting because other folks have found that elevated levels of variation in regulatory genes are correlated with morphological diversity. So of course, this is like a preliminary finding. I'm not saying that genotype is and phenotype, there's a nice and easy story here. But in a group of fishes that's known for their weird cranial anatomy, something strange is going on in the regulatory gene that has something to do with the ontogeny of those cranial elements. So to sum this up, we got a pretty good tree for Ballistoidea. We did find several genera that were non-monophyletic that will eventually be in need of taxonomic revisions. BMP4 evolved particularly quickly in filefishes. It does have phylogenetic utility. It did contribute pretty significantly to our structure. However, you kind of got to use it with a little bit of a grain of salt because we did run into some long branch attraction issues when we looked at a broader taxonomic sampling. It's also time to go next gen with this group. There's this constant want to kind of apply a finer focus to the phylogenetic resolution of tethered oniform fishes. And I know in our lab we're certainly looking into that, and I know other folks are as well. And also, really, we got to get down to this part of the world and sample the filefish biodiversity because I think there's a really interesting story there. And we just don't have the choo-choo samples from that part of the world yet. So we've got this tree. Next, I wanted to use it to analyze the cranial diversity of triggerfishes, which is a much smaller group than filefishes. I was able to essentially look at the anatomical diversity a lot easier in this group. So, don't worry about too much of the details here. Triggerfish schools are crazy. I promise you can come and talk to me later about it all day. We're going to be coming up with a publication about this in the near future. The takeaway is that they do look different from a typical teleost in a couple different ways, several of which are listed here. Now, you'll notice that I have named these mussels using names that you might not be too familiar with. You're probably more familiar with this alphanumeric nomenclature. However, I've decided to use the names more recently proposed by DeTovo and Berry because they're a lot easier to apply to a complex musculature, such as what you see in triggerfishes. So, essentially, although all triggerfishes look very different from another fish you're going to pull off the coral reef, there's this idea that they're a very homogeneous group with respect to cranial morphology. I couldn't help but question that and wanted to figure out where the biodiversity was. And so I used geometric morphometrics in conjunction with phylogenetics to investigate that a little bit further. So just as a reminder, this is the triggerfish portion of the maximum posterior probability tree from the first part of my talk. This has been pruned for triggerfishes, obviously, and so we're using this topology and branch information for everything you're going to see here in a minute. This is a morphometric landmark protocol that I came up with to describe the shape and also outline jaw joints where tendons and ligaments are, also outline the shape of muscles. So the idea is to not only kind of capture what the overall triggerfish skull shape is, but also outline some potentially functionally relevant elements. So I dissected a bunch of specimens from 27 different species, all museum collections, subjected them to a Procrustes fit, and then translated them into morphospace. And before I get into my PCA analysis, which even just in this one little slide, there's so much crazy stuff going on, I wanted to say that I found that both size and phylogeny were significantly influencing the shape patterns that I found. So everything you're going to find or see from here on out includes a phylogenetic component, and also I'm using the residuals of a regression that I did of shape and size. So again, this is a PC plot. Essentially, each of these points is each species' average cranial shape that has been projected into morphospace, and then I've connected it using a phylogeny. So here's the root of the tree, and you can kind of see from this root there was kind of this massive diversification, and then once these different clades kind of reached the limits of that, they kind of oscillated within there. You're also going to see that different clades have invaded, obviously, different portions of morphospace, but also different, I guess, areas within it. So like clade A, or clade 1, for example, is limited to the small little portion up here, whereas clades 2 and 3 have kind of spanned out a little bit more. There's also lots of lines crossing over each other and spreading out, so that's pretty interesting. That's indicative of shape diversification or convergence. There are some portions, like here, this is Rhinacanthus and Supplement, two major genera within the group, where genetic distance and shape distance is about equal. In fact, they are the only group that is in the positive portion of PC1 morphospace, except for this little critter here, Ballisticus undulatus, which is converging on that shape space. There's also significant examples of shape divergence amongst closely related organisms. Up here, we have Balissoides viridescens, which is in a totally different region of morphospace as compared to its close relatives within the genus Xanthipes. So ecology seems to also be giving some structure to this. Generalists here, I've essentially recoded those points based on meaning ecology. There's three main ecomorphs. Generalists span PC1. Kind of makes sense. If you're eating all sorts of different things, there's probably many different ways you can look. Whereas the trophic specialists are kind of a bit more limited in the portion of phylomorphospace they're invading. And interestingly, PC2 seems to actually pretty nicely divide the Durophagous triggerfishes from the algae eaters. Based on some statistical analyses, I also found that the shape distance between each of these three feeding gills was significant. Also, in this case of divergence that I talked about before, relative to these closely related organisms that have splits, in this case, this seems to be potentially due to feeding ecology. These fishes that are closely related genetically have diverged from one another in order to converge upon the shape space that is occupied by fishes of the same feeding order. So I'm going to kind of finish this up. Of course, there's shape features that we can look at here, but the takeaway is that both the overall kind of shape of triggerfish skulls as well as the functionally relevant aspects of the triggerfish cranium have changed over time. And the functional morphologist in me could not help but ask, what are those functional significance? And of course, that was the other part of my dissertation research. I'm not going to get into it right now, but my landmark protocol I've made in order to be biomechanically informative. Essentially, we could use this information based on the relative position of muscle insertions, their lengths, jaw joints, and we built a jaw biomechanical model that tells us all sorts of really interesting information about what the bite performance of triggerfishes is. So we already kind of talked about this. Really fantastic group, along with their relatives, to put together a great evolutionary story talking about the evolution of form, function, and ecology. So first and foremost, thanks to my boss and advisor, Mark Westmead, Dr. Xiao in Taiwan, who let me collect the majority of the species that I used to sequence the data to form my phylogeny. And also, I'm here kind of with the Future Phi project. Come and see our booth. Tell us about what your priorities are as the fields of biodiversity and phylogenetics move forward. So again, I will be at the booth, but other ways to get in touch with me, and I would be happy to take any questions.